As he said, my name is Alex. I'm a PhD student in Gioele Manno's lab at EPFL, and I'm going to discuss the RNA velocity advancements we've been working on in the lab, specifically for the cell cycle and other periodic manifolds, and it's going to complement the talk you heard earlier. Just in case you weren't there, I'll give a little bit of recap on RNA velocity. Essentially, the idea is that we can take advantage of the fact that in single-cell RNA sequencing data, we capture both unspliced, or U, and spliced, S, reads for the same gene, and that there's a temporal component to their expression in the cell. In that first, we have transcription of a particular gene into U, followed by a splicing of that U into S, which exists in the cell for some time until degradation, according to some rate represented here by gamma. And we can take these two data quantities as well as the rates and represent them in a series of differential equations, which I show here on the bottom. In particular, this equation here, which represents the change in spliced over time. And this is a function of the rate at which the unspliced is spliced, according to beta, minus the rate at which the S is degraded. And once we estimate this on a gene-wise matter, we use phase portraits to estimate a residual from a steady state fit and project this onto a low-dimensional space, such as a UMAP, which I show here on the right. In this case, I'm showing some single cells that are undergoing the cell cycle. So they're progressing from G1 to S, G2, and M. And likewise, the velocity suggests that there is a progression in gene expression space of those cells along this known biological process. However, there are several limitations to the existing velocity frameworks that are out there. First is the fact that they rely on K nearest neighbor imputation to smoothen the data and overcome data sparsity. Additionally, we're a bit limited in whether we can perform reliable and robust statistical inference, particularly between different conditions. Most importantly, velocity is inferred on different timescale for each gene, and since velocity is typically represented in a low-dimensional space averaged across all those genes, this creates what we call the consistency problem, where this low-dimensional velocity effector field is no longer consistent with the real high-dimensional gene expression manifold. What do I mean by a gene expression manifold? So this is the high-dimensional representation of the many genes you measure with single-cell RNA sequencing, as well as a slightly lower dimensional representation describing all the possible biological states through which your cells may traverse. And here I'm showing an illustration of this manifold where each dot represents a cell, and these cells must lie on this gene expression space. And likewise, we would anticipate that the velocities should also remain tangent to that manifold. However, in practice, since we have this different scales for each genes, we do not preserve this consistency, which led us to ask whether we could write a system of differential equations for RNA velocity that links the manifold and the velocity together, thereby creating a consistent velocity. And if we want to do this, we also need to ask what is our true gene expression manifold, because the problem is that we have very complex processes that don't unfold on a low-dimensional space, such as differentiation and development, and therefore it's much more difficult to define what our manifold is. So we decided to implement our theory for a low-dimensional case that is very pervasive in single-cell data, and that is the cell cycle. The cell cycle is a one-dimensional circular or periodic manifold, which I represent here with this torus, where a cell has a position on the manifold, which can be considered an angle, phi, and we want to reformulate the RNA velocity equations for this scenario. So we take into consideration the fact that we have a manifold, Y, and each cell is going to have a position on that manifold, which we call X, and this is composed of the information we get from single-cell RNA sequencing, so our unspliced and our spliced counts. We can then take the differential equation I showed in the first slide, and we can perform the chain rule to get the derivative, and from that, we can plug it in to the equation, where now the velocity is directly a function of the position of a cell on the manifold, or x. And in our case, since we're dealing with a periodic manifold, we can replace x with phi, which corresponds to the phase of the cell cycle. And in this case, we also consider the velocity to be a speed, since we're talking about a one-dimensional space, and we represent this with omega. We wanted to implement this in a way that would allow estimation of uncertainty so that we can do more statistical comparisons. And for this, we designed a probabilistic model, which I'm showing a simplified version of on the right. And essentially the way this works is that each variable is drawn from a different distribution. So we expect our data, spliced and unspliced counts, to come from a negative binomial. And likewise, we have various different parameters, including the degradation rate and the splicing rate, which also come from their own probability distributions. The question is how we got to this from the equations I showed in the previous slide. And this is simply a reformulation of the last equation I showed on the slide, so that we get the U term on its own on the left-hand side of the equation. And the key here is that since we're talking about periodic manifolds, we're representing this as a Fourier series, so a series of cosine and sine terms that describe periodic processes. And we do this for both the velocity itself as well as behaviors of the genes, or the gene harmonics. Here I'm showing a plate diagram representation of our model, which we solve using variational inference. And we solve it in two steps. In the first step, we perform phase inference, so we learn the manifold. And this is represented in this diagram here, where the gray circles represent our data points, and the white circles represent our random variables, each of which is sampled from a different probability distribution. And the arrows represent the relationship that they have with one another based on the series of differential equations that define RNA velocity. So first, we learn the cell cycle phase, as well as some other parameters that describe the behavior of genes along the cell cycle. And then we condition on that information we learned in our second step and perform estimation of the velocity, which is a function, omega, as well as the velocity kinetic parameters, such as the splicing rate and the degradation rate. Now, in the last minutes of my talk, I'm going to show some plots to answer the important question we wanted to explore about the posterior estimate, and this is namely whether the model preserves relationships among the variables that we expect to have some sort of relationship with one another, given the biological formulation of the problem. So for example, here I show two plots where each dot represents a gene, and this is the estimate of those genes after running our model. On the x-axis, we have the splicing rate, or beta, and on the y-axis, we either have the total spliced counts on the left or unspliced counts on the right. And what we see is that the splicing rate that we estimate is positively correlated to the total expression of that gene in terms of the spliced counts. However, in terms of the unspliced counts, there is an anti-correlation. And this makes sense biologically because if we have a very fast splicing rate, we expect there to be little unspliced because it's quickly processed into more mature spliced RNA. Likewise, if we look at the estimates for gamma, the degradation rate, which I show on the x-axis, we see that there's a correlation between the degradation rate and the splicing rate for many genes. So here, again, each dot represents a single gene. We also see that if we estimate the shift between our expected unspliced and spliced counts, so this is the fit and the delay along the cell cycle, we see that there's an anti-correlation with the degradation rate, which in other words means if we have a faster degradation rate, we have less delay between our unspliced and spliced, which is a way of visualizing the velocity. Last but not least, we wanted to see if another approach besides variational inference, namely MCMC, would provide us more insights into the underlying structure of the data. Namely, if we look at the uncertainty of our samples from MCMC for different parameters, such as the degradation rate and the velocity, we expect either two scenarios, one where there's a correlated uncertainty, or where there is an uncorrelated uncertainty. And what we found is that for different genes, we have either a correlated or an uncorrelated uncertainty. What we think this suggests is that for some genes, we're able to elucidate whether there is a contribution to the velocity or not, and that this tells us which genes hold velocity information. And we want to take this information back to our variational inference implementation and perhaps represent these parameters in a multivariate normal variational distribution instead of just a normal distribution. To summarize, RNA velocity vector fields are inconsistent in their current form with the gene manifold, so we took an approach where we link the manifold and the velocity together in a more heuristic-free way, and we applied it to the cell cycle and periodic manifolds. With that, I'd like to thank everyone in the lab, and most importantly, you for your attention. And I hope to see you at my poster if you have any further questions about single cell or velocity. Thank you.